Alrighty, Science 10s here. We are going to be talking about transpiration. So this is actually something you can do at home. You can just take a stalk of celery, put it in some food coloring, and determine some different factors that are going to affect the rate of transpiration. So I'm sure you can think of a lot of different environmental factors, right? I mean, if you start thinking about transpiration, the movement of water up the xylem from the roots to the shoots, you can probably think wind, right? Wind might affect the rate of transpiration. What other things? Whether there's leaves or no leaves on your celery plant, whether you're exposed the plant to sunlight, spelled wrong, sunlight, or leave it in the dark. What else could you do? Temperature, right? What are the effect of temperature? High temperature versus low temperature, right? There's all sorts of different factors that you could look at and study. And so we can easily do this at home, right? If you have a fan at home, there's wind. Leaves or no leaves, right? Cut some leaves off compared to one with leaves. Sunlight, place in front of a windowsill, dark, in a cabinet. Temperature, heat lamp, or putting it in a warm place beside a heater as opposed to a cool place out in the garage or something, right? So there's a lot of different things that you can do here to decide a factor that would affect the rate of transpiration. So you can come up with a manipulated variable. What are you changing? Okay, so what are you going to change? And then obviously it's going to be the responding variable is going to be the rate of transpiration. So how do you think we should measure that? Well, of course, there's a couple ways of doing this. We could measure the mass of the plant before and after. We could measure, put a plastic bag around it and look at how much moisture is being released. Another thing, we can simply use food coloring. Put some food coloring in the water, and over time, we expect that it'll be drawn up the stalk of the celery into the leaves. Then you have to think about what needs to be controlled. What do we have to keep constant? Which things need to be kept the same? Because obviously, if we change them, they would change the responding variable. So we can come up with a hypothesis, right? Once you and your group mates figure out what the manipulated variable is, if manipulated variable, then responding variable because scientific reason. So an example might be if we add a fan to our celery plant, then the rate of transpiration would go up because, well, why? And then talk about, in terms of your scientific reasoning, what do you want to talk about? Well, not just transpiration, but why does transpiration occur? So you should use words like cohesion and words like adhesion and differences of osmosis, right? Water moving from a high concentration to a low concentration. And think about the plant. How does water move through the plant? Where does it go? And think about that. It starts at the root push at the bottom, and then there is going to be the transpiration pull at the leaves. And don't forget about the leaves, those stomata, right? Those little holes, the little openings on the underside, of the stomata. That is something that is going to affect the amount of transpiration. We can kind of think of transpiration as like the rate of evaporation, right? Is when the stomata are open, water can leave the stomata, and that it evaporates out of the surface of the plant. So one way to quantify, to actually measure the rate of transpiration, scientists can use this little thing called a potometer. And so here's what it is. It just looks like this little, I don't know how you want to explain it, little stalk, and there's some water in here, and we're going to put a plant just here. And then here are the different factors that we can look at in this particular study. So there's a little heater here. Here's a fan to mimic or simulate wind, and here's a light as if it was sunlight. Okay, and looks like we're keeping the room temperature constant. Okay, so temperature constant in the room. And we're going to select a different plant. We've got 4 different plants here. I'm just going to randomly grab one. And I'm going to put them into the little apparatus. So I've chosen a zebra plant. And we're now going to measure using this little device how much transpiration is occurring in like a little fake hour. So here's my little timer. Here's like a fake hour. Oh, look at that. The zebra plant transpired 4.2 mils. Okay, so that's our baseline. That's our control, right? And so now we're going to take a look and decide if these factors are going to increase the amount of transpiration or decrease the amount of transpiration. So I'm going to be adding now some heat. And well, let's see what's going to happen. Like, let's think. What do you think? Do you think transpiration will go up or transpiration will go down? Okay, so you think about that, and then I'm going to start the timer. There we go. Okay, so 6.1 mils. So that went up by adding our little heater. So what might be the reason that the transpiration rate went up? Why was there more evaporation of water? Why was there more transpiration? We'll compare this to a fan. This is going to mimic or simulate the effect of wind on this plant. Okay, so there we go. Oh, look at that. Well, 7.6 mils. That's almost doubled. So certainly that increased the rate of transpiration. And now we can compare to adding a lamp. It's like mimicking sunlight. There's our little lamp is on for our fake hour. And look at that. The amount of transpiration went down. So kind of makes you wonder between the control rate of 4.2 mils and this went down to 3.2. I'm almost wondering if maybe the light had the stomata closed, right? Like what happens on a hot day with stomata is they will close so that they don't lose too much water. So you have to wonder if maybe that's what's happening here. So now I'm sure we could repeat this. There's all other different plants that we could try this with, geranium and ivy and weeping fig. So understanding that no matter what kind of plant we're having, the mechanism is going to be the same, right? Is that transpiration occurs from the root with the root push and the water moves up the xylem to the leaves of the plant. It's going to move out the stomata. Why? Because there's a transpiration pull because of the difference in the concentration of water between the plant leaf and the outside external environment. And then we can mimic this, of course, using these different test conditions or these different manipulated variables. And so that's exactly what we're looking at in this lab. So you guys will take a stalk of celery, cut the ends off, fill some water into a plastic cup, add food coloring, and then apply your particular manipulated variable. What treatment are you using? Are you going to be the fan imitating wind? Are you going to try temperature? Are you going to try leaves, no leaves? What are you going to do that is going to change the transpiration rate? And then we will talk about it. We're going to look at, for our responding variable, the change in mass as a way of measuring the amount of transpiration. And I said there's other ways of doing that. How much water was transpired? You could record that using the little device that we saw. Okay, and so we'll go through and be able to answer these questions based on you doing your lab. Okay, that's it. That's our transpiration lab. All done.